Welcome back to Chapter 6, Enzymes. In this lecture, we are going to look at the practical applications of enzyme kinetics. This lecture is going to help you understand the quantitative analysis of enzyme function. In addition, this lecture will also help you to comprehend enzyme inhibition, how inhibitors work. That is one of the main topics of this chapter. So we talked about in great detail the quantitative description of enzyme kinetics in the previous lecture. Now what I'm going to do is to try and make you understand the applications of that quantitative description. Now, application comes in various stages. The first application is when you perform an experiment in lab. All right. Now, when you perform an enzymatic reaction, when you use an enzyme and a substrate, how would you perform this reaction? And for this, we normally measure the initial rate of the reaction. The initial rate is given by V0 of an enzymatic catalyzed reaction. Conditions for measuring the initial rate usually entails a large excess of substrate compared to the enzyme. So what this means is that when you're doing an experiment wherein you're converting a substrate to a product using an enzyme or performing an enzymatic reaction, you have to have a large excess of your substrate as compared to your enzyme, concentration wise. Normally, we fix the enzyme concentration and measure initial rate at a variety of substrate concentration. This means that we do various assays, and the difference between these different assays is the difference in the concentration of the substrate. The concentration of enzyme is constant. Eventually, changing the substrate concentration will have little or no effect on V0, which means we have reached the maximum velocity of this enzyme, or Vmax. Recollect the term Vmax. And Vmax is an important term because it describes the maximum velocity of an enzyme, above which the velocity cannot go. So let us look at an example. How to do kinetic measurements. And here is an experiment. First, mix enzyme and substrate. Now, what sort of enzyme are we talking about? Let's take an enzyme called as alkaline phosphatase. An alkaline phosphatase, what it does is it hydrolyzes monophosphates. And a substrate that we are going to use is nitrophenyl phosphate. And when you treat it with alkaline phosphate, alkaline phosphatase, what you're getting is just a phosphate plus nitrophenyl ion. So what it is doing is it is cleaving this bond and it is adding an oxygen to the phosphate. So this is P double bond O. Now this is a very important reaction. This is an example of alkaline phosphatase. Now the way we monitor this reaction or record rate of substrate disappearance or product formation as a function of time is using absorbance. So we are measuring absorbance over time. Now, how are we measuring absorbance is the nitrophenyl product that is coming out of this reaction is can be monitored at 400 nanometers. And what happens as the enzyme converts nitrophenyl phosphate to just phosphate and nitrophenol, you can see that the reaction increases. You see, as you increase the time, the absorbance goes up, right? Goes from 0 to 1, for example. Now, if you do this at one fixed concentration of a substrate and enzyme, right, you would see this one curve. Now, if you decrease, if you do this at lower substrate concentration, you will have these kinds of curves. Now, what you're looking for is the initial rate. So the initial rate is this portion. Now, this is still, let's say this is about 100 seconds. 100 seconds. And the total time is, let's say, 1800 seconds. And at 100 seconds is the initial rate, or this is given by V0. And that's what we are measuring. Plot initial rate versus substrate concentration. Change substrate concentration and repeat. Now, if we increase the concentration of the substrate, as you can see, the initial rates change. In this case, the initial rate is increasing. So let me redraw this curve for you. The curve is absorbance versus time, right? Absorbance at 405 nanometers or 400 nanometers versus time. And what you need to do is to take the initial rates, just the initial rates until here. And this, from this initial rate, you will obtain the concentration of the product. So the concentration of the product can be plotted separately versus time. And in this case, the product is nitrophenol. Right? Now, these are the two values, the initial rate and the substrate concentration that we need for Michaelis Menten plot. So if you have a plot of product concentration versus time, as shown here, you can get initial rates from this and then plot it separately, where you plot the initial rate on the y-axis and substrate concentration on the x-axis. And if you do so, you'll get various points. And these points can be fitted to a curve. And that curve is shown here in black. I'm redrawing it. And that curve is obtained by fitting these red points to the Michaelis Menten equation. Right, Vmax S over Km plus S. And from this, what we obtained is the value of Vmax and Km. So the Michaelis Menten plot, and you obtain Vmax, which is the maximum rate as shown here, right? Km, which would be the substrate concentration, where you have half saturation. Now, remember, the specificity of an enzyme is given by kcat divided by Km. Now, what you obtain from this plot is Km and Vmax. And Vmax, as you know, is equal to kcat times E0. So kcat is equal to Vmax divided by E0. E0 is your substrate concentration. Sorry, E0 is your enzyme concentration. So you know the enzyme concentration because you calculated the enzyme concentration and you did the assay. So you divided the Vmax, we divide Vmax by E0, you get Kcat, and you have Km, and then you can calculate Kcat on Km. This is how you measure the activity of an enzyme. The Michaelis Menten equation can also be rearranged in such a way that you take a reciprocal of this equation, as shown here. If you take the reciprocal of this, is 1 by V0. Now, you can rearrange this equation again and obtain 1 over V0 is equal to Km divided by Vmax times 1 over S plus 1 over Vmax. And this is very similar to our straight line equation, right? V is equal to mx plus c, where your y is equal to 1 over V0, x is equal to 1 over S, and your c is equal to 1 over Vmax. So the known values are V0. You obtain V0 from your absorbance values, and S is the substrate concentration that you used in your assay. So you can use this reciprocal equation to plot 1 over V0 versus 1 over S. So if you plot this reciprocal equation, what you get is this inverse plot or double reciprocal plot, and it is linear. Now, if you get a linear plot like this, you can get Vmax and Km from this. The way you get Vmax and Km is by looking at the intercept, the y-intercept. As you can see, this is c, right? Y equal mx plus c. C is 1 over Vmax, and slope, or m, is equal to Km divided by Vmax. And the intercept at the x-axis is given by minus 1 over Km. So this itself will give you the value for Km, and the intercept will give you the value for 1 over Vmax, and slope will give you value for Km over Vmax. And from these values, all these values, you can get Kcat, right? And then you can calculate Kcat on Km. So these are two different variants of Michaelis Menten equation. Lineweaver and Burk first obtained this kind of a double reciprocal plot because in those days, plotting a nonlinear least square plot was extremely difficult. Computer programs were not accurate, although nonlinear least square fits are statistically more accurate as compared to linear plots. Enzymatic reactions can also involve two substrates, which can get converted to two products or one product. Let's look at an example. Let's say you have an enzyme, right? And this enzyme can recognize two substrates. Let's say A and let's say B. Now, this enzyme fits these two substrates in the active site. Perfect. So the function of this enzyme is to combine these two substrates. And what you get is a product. Plus A, B. So if you look at this, there are two substrates, A and B, that is bound by the enzyme, and then it joins them together to form AB. Now this is a two-substrate reaction leading to one product, right? Now this kind of a substrate product reaction can be written as E plus S1, S2, giving to ES1, S2, giving rise to E plus P, right? So how do we deal with this kinds of reactions? The question that one may ask in this kind of two substrate reactions is, how does the enzyme recognize both substrates? Does it recognize A first or B first, right? For example, now let's redraw the enzyme, right? And you have two substrates, A, B. So it is important that we understand the mechanism by which this enzyme recognizes these two substrates. And that is called as a kinetic mechanism. The order of binding of the substrate and release of products is what this mechanism entails. That's what the mechanism tells us. And why do we care, and why do we have to know that? It is important for us to know because the way enzyme recognizes one of the substrates can dictate as to how this enzyme functions. Now, there are many different enzymes that performs two substrate reactions. And the mechanism of recognition of substrates by these enzymes are different in different cases. So when two or more reactants are involved, enzyme kinetics allows us to distinguish between different kinetic mechanisms. There are two different kinds of mechanism. One is a sequential mechanism, the other one is ping pong mechanism. As you may have guessed, it comes from the game ping pong, right? Ping pong is played when the ball is alternated between two players. So it goes to the next court, comes back to your own court, right? So let's look at these two mechanisms. So two different kinds of mechanisms. One is sequential, where the enzyme reaction involves a ternary complex. And a second a mechanism where the enzyme reaction does not involve a ternary complex. Now let's look at the first one. And let me define as to what a ternary complex is. As you can see, ES1, S2, which means that the enzyme is binding both the substrates at the same time. In a mechanism where there is no ternary complex, this kind of a complex is not present. The way this complex is formed can be via two different mechanisms. It could be random, which means that E can first form ES1, or it can first bind S2. So it can either bind S1, the first substrate, or the second substrate, and then form this. And then it can release P1 and P2 in this case. Ordered, which means that it binds the first substrate always, and then binds the second substrate, forms a ternary complex, releases the products. This kind of mechanisms is not directly applicable to the example that I showed you, where two substrates bond, one product come out. That was just an example that I told you. In this case, you have two substrates and two products coming up. In the ping pong mechanism, which is very easy to remember, is that the enzyme binds one substrate, okay, and then releases a product. It binds another substrate and then releases another product. So it's a very easy thing to remember. But how do we know that the enzyme is doing either of these mechanisms? Right? It has to do either of these. It's not going to do both mechanisms. That's not logically possible. Let's look at some graphs. Now, how does a sequential mechanism look like? And shown here is a Lineweaver-Burk plot, or an inverse plot, right? So this is an inverse plot shown here, 1 over S versus 1 over V, as shown here. And you can see these lines, and you know what this intersection means, right? The slope means Km over Vmax, intersection means 1 over Vmax, and stuff like that. Now, if you do this, now the way this experiment is done, unlike the original one substrate, one reaction experiment, in this case, what you're doing is keeping the enzyme concentration constant and keeping substrate 2 or S2 concentration constant, and you're varying S1. S1 is varying, and hence you get lots and lots of concentrations of S1. In the second experiment, you're keeping the enzyme constant, you're varying S2 to a different concentration and changing the S1 concentration, and hence you get various different concentrations of S1 and henceforth. So this explains it. You vary S2 concentration. For each experiment, S2 is constant, but for different experiments, S2 is varying. Now, when you do this experiment, and when you have the intersection either on, when you have any sort of intersection of lines, intersection of these lines, either at y-axis or behind the y-axis, lines intersect. One of the most important things for this mechanism is that lines intersect, and hence a sequential mechanism. Now, the caveat is that it is extremely difficult to differentiate between random mechanisms, random and ordered mechanisms, because even though random mechanisms in equilibrium will give intersection at y-axis, that rarely happens, and so it's extremely difficult to differentiate between random and ordered. What you can say is it's a sequential kinetic mechanism. The ping pong mechanism, on the other hand, is really straightforward to understand. Using Lineweaver-Burk plot, if the lines do not intersect and are parallel, then it's ping pong kinetic mechanism. So in this way, one can use enzyme kinetics to differentiate between different kinetic mechanisms. Enzymes can be inhibited. Inhibition of enzymes slows or stops the catalysis, which means that the substrate will not get converted to product. Inhibition can be reversible or irreversible, which means that in a reversible case, the inhibitor can leave the active site of the enzyme, and it's always in an equilibrium. Irreversible means that the inhibitor makes a covalent bond with the enzyme, and it will not leave the enzyme. Reversible inhibition can have different mechanisms. It can be competitive, uncompetitive, or mixed. Non-competitive is a special case of mixed inhibition. Let's look at the first case, competitive inhibition. What this means is that if you have an enzyme that is shown here, E, and it has a binding site for the substrate that is shown here, and if substrate is this hexagon and an inhibitor is this pentagon, remember the inhibitor can occupy the substrate site. This is what it means by competitive inhibition. The inhibitor competes with the substrate for binding. And that's what is written here as well. It binds the active site and does not affect catalysis. Now, What this means is that there is no change in Vmax for competitive inhibition. It increases the Km for a substrate. Let me explain it to you. Now, this is a classic enzyme substrate reaction, E plus S going to ES going to E plus P. Now, if there is an inhibitor that competes for the substrate shown here, the scheme changes, and the scheme changes like this: E plus I going to EI. Because this is a reversible inhibitor, as you can see, it's a reversible arrow here, right? And this is dictated by an equilibrium constant called as Ki. This is a big Ki. This is an equilibrium constant. Now, what it means by apparent change in Km, it increases the Km for the substrate. Now remember, Km is the ability of this enzyme to bind a substrate. It directly relates to the binding affinity of a substrate. Now, if an inhibitor binds to the same site as a substrate, it means that the inhibitor, the enzyme, cannot bind the substrate with the same affinity. Yes, that is why it means apparent increase in Km. So a competitive inhibitor increases the Km. However, it does not increase Vmax or Kcat. Quantitatively, a competitive inhibitor changes the Michaelis Menten equation. Now, in the original Michaelis Menten equation, there is no alpha. When you deal with a competitive inhibitor, it introduces a term alpha to the Michaelis Menten equation. And alpha is given by 1 plus I over Ki. One can actually derive this equation from this scheme using steady state approximation. But for this course, you are not required to do that. What you're required to do is to remember that alpha involves this particular term. And it means that because there is an alpha attached to Km, that means that Km of that enzyme will go up. And Ki, that is shown here, is given by the concentration of A times I divided by EI. And this is called as a Kd of the inhibitor. Graphical representation of this competitive inhibition. And as you can see, you can use an inverse plot to figure out the differences between various kinds of reversible inhibitors, whether it's a covalent inhibitor, non-covalent inhibitor, or an uncovalent inhibitor. Now, as you can see, this term alpha is embedded in this equation. Now, if you were to increase the concentration of inhibitor, all right, in each experiment, remember each experiment is constant enzyme concentration, variable substrate concentration. Now, in this case, if you also involve an inhibitor at various different concentration, or one concentration of inhibitor where you are varying the substrate concentration, and you can vary the inhibitor concentration, and hence the curves or the lines are different. And when you get these lines, and if the lines intersect at the y-axis, this is a characteristic of competitive inhibitor. Now, remember, C is 1 over Vmax. If they intersect, that means that Vmax is not changing, whereas Km is changing because Km is the intercept at x-axis. And as you can see, Km is given by minus 1 over alpha Km. And yes, the alpha Km is the apparent Km. And it changes. So competitive inhibitor increases Km. No change in Vmax. And this is the quantitative description for this phenomenon. The next type of an inhibitor is an uncompetitive inhibitor. So an uncompetitive inhibitor is something that binds in a site away from the binding site. For example, if your substrate is a hexagon and your inhibitor is a pentagon. However, this does not bind at the same site as the substrate, and it's binding at a site that is away from the substrate. Now, the way you can actually explain this in a scheme is this. The inhibitor is not binding the enzyme. That's the most important thing. This inhibitor is not binding the enzyme. The inhibitor is binding the ES complex. Once the enzyme and substrate encounter and form the ES complex, the inhibitor binds that complex. And you can incorporate and change the scheme in such a way that ES goes to ESI in a reversible equilibrium. And this is given by Ki prime. Now, let's look at some characteristics here. The inhibitor only binds to ES complex, does not alter substrate binding. That's important. It does not bind substrate binding, but inhibits catalysis. Cannot be overcome by increasing substrate concentration. Whereas competitive inhibitor, if you increase the substrate concentration, you can overcome the effects of an inhibitor. What else is happening? Decreases Vmax. Apparent decrease in Km, but no change in Km over Vmax. So there are a couple of things that you need to understand. Is that in an uncompetitive inhibitor, it decreases the Vmax, and there is apparent decrease in Km happens, but it does not change the Km over Vmax. Now, how can you explain this quantitatively? Uncompetitive inhibition is classically seen for enzymes with more than one substrate, and it binds to ES complex. Now, in this case, the alpha is associated with the substrate. And so the alpha term, or alpha prime, is given by 1 plus I over Ki. And in this case, a Ki prime is concentration of E times I divided by EI, which is the same. Even here, the Michaelis Menten equation changes in the presence of an uncompetitive inhibitor. Let's look at how the graph changes. For an uncompetitive inhibitor, the Lineweaver-Burk plot, or the double-reciprocal plot, is parallel. This is such an easy way to remember, right? Now, if you look at the double reciprocal equation here, the alpha prime is on Vmax. So this is the intercept term, right? So there are various different y-intercepts, as you can see, and x-intercepts. So alpha prime over Km changes and Vmax changes because Vmax different slopes here, and ultimately Vmax is decreased. Vmax goes down and alpha Km decreases. So because if you look at it intuitively, if you're increasing the inhibitor concentration, 1 over V0 is increasing or V0 is decreasing. The same way, 1 over S in this case is decreasing, as you can see here, right, going there, or S is increasing, but at the same time minus alpha over Km is this term here is decreasing, and alpha minus alpha prime over Km here. So you have to remember Vmax goes down in uncompetitive inhibition. Mixed inhibitors are different than competitive and uncompetitive inhibitors in the sense that mixed inhibitors bind enzyme with or without substrate. So as you can see here in this diagram, this is an enzyme E, and an inhibitor can bind to the enzyme from the EI complex, right? If this is the original scheme and if this is the inhibitor scheme, right? Now, this inhibitor, this EI complex can then bind a substrate from ESI, or the enzyme can form an ES complex first and the inhibitor can bind to form the ESI complex. So this inhibition is a classic case where it doesn't show any bias towards binding to enzyme or the substrate. Now, a characteristics of this mixed inhibition is that there is decrease in Vmax and apparent change in Km. So mixed inhibition is like a combination of competitive and uncompetitive inhibition. So there is a decrease in Vmax and also apparent change in Km. A special case of mixed inhibition is called as non-competitive inhibition, or inhibitors that follow non-competitive inhibition are called as non-competitive inhibitors. They are mixed inhibitors with no bias towards EI or ESI state, thus show no apparent change in Km. Now, the way you can differentiate in a plot is in Lineweaver-Burk plot, lines intersect left of the y-axis. Let's see how that happens. For a mixed inhibition, the Michaelis Menten plot or the Michaelis Menten equation changes in this format. Km has got a term alpha. This means that apparent Km changes. And in the denominator, the concentration of substrate has another term alpha prime. So the equations are pretty similar to what we have seen before. Non-competitive inhibitors are those that have alpha is equal to alpha prime. And as I said, this is a special case of mixed inhibition. For mixed inhibitors, or in case of a mixed inhibition, the Lineweaver-Burk plot or the reciprocal plot looks like this. And there is intersection, but the intersection is on the left-hand side of the y-axis. And as you can see, Km, apparent Km changes because minus 1 over Km dictated by alpha prime over alpha times minus 1 over Km is changing. Also, alpha prime over Vmax is changing because the intercept is changing, and hence Vmax is decreasing, Km is increasing. For non-competitive inhibitors, as I said, alpha is equal to alpha prime, and so no change in Km. Therefore, the x-intercept would not change on a Lineweaver-Burk graph. So here are the three plots shown in a same slide. On the left-hand corner is a competitive inhibitor. Now, as you can see, lines intersect at y-axis, and so there's no change in 1 over Vmax, and so Vmax doesn't change, whereas apparent Km increases. In case of an uncompetitive inhibitor, the lines are parallel, no intersection, and hence Vmax decreases, and there is slight change in alpha over Km. For mixed inhibitors, the lines intersect on the left-hand side of the y-axis. As a result, there is change in the intercept and hence Vmax decreases and apparent Km increases. Please remember the difference between these three inhibition types. Irreversible inhibition is another type of inhibition that involves covalent attachment of an inhibitor to an enzyme. An irreversible inhibitor kills an enzyme's activity. It can form a covalent bond with an active site residue, and it happens in most of the cases. Protein toxins often act by irreversibly inhibiting cellular components. An example is diisopropyl fluorophosphate. It's an inhibitor of the enzyme acetylcholine esterase. Diisopropyl fluorophosphate is a potent neurotoxin. The way it works is that acetylcholine esterase has a serine hydroxy that is important for its activity. DIPF with this structure, it's a fluorophosphate, as you can see, and this bond is electrophilic, or it's very reactive. Phosphorus being an electrophilic center, the hydroxy attacks this phosphorus, and as a result fluorine leaves, right? And if you were to write a mechanism for this, you could write in this way. So it attacks and fluorine leaves, and you are left with the enzyme inhibitor complex. This is an inactivated enzyme. Transition state analogs are different from reversible and irreversible inhibitors. They're quasi-irreversible because they have a higher affinity for enzyme active site than substrate. For example, in case of a proline racemase, the reaction catalyzed by this enzyme is racemization of this L-proline to D-proline, and a transition state involves a planar structure. Inhibitors such as pyrrole 2 carboxylate or delta 1 pyrroline 2 carboxylate, which have a planar kind of a configuration, can mimic the planar transition state of this reaction. And hence, these two compounds can become inhibitors of proline racemase.